Hello, welcome everyone. It is Thursday and it is that time again for our webinars. Every week we do engaging webinars on science, neurodiversity, and better understanding our own brain. This year we've had a massive deep dive into neuroscience, looking at how its impact on neurodiversity, where it is pushing us forward, and where we are currently being held back. We are almost at the end of the year and we are now looking CRISPR. If you've never heard of it, you're in for a treat. It seems like science fiction, but it is actually happening right now. And we're also going to be looking at, does the neurodiversity movement and the idea of genetics and altering genetics, can they be one, or are they always going to be butting heads? So those are the type of things we are going to be looking at. April, autistic, wants to learn more about DNA and gene editing, and I'm here to record. Thanks. Ooh, April, neurospicy. Mixed feelings about the subject, so I want to know more. And do you know what, Tracy? That's completely understandable to have mixed feelings about it. I would say technology is advancing whether we like it or not. So it's better to be informed and part of the discussion because the rules and the regulations are being created as we speak. Okay, research on this, children nice, have a background. Oh, greetings. Hello, everyone. Lovely to be here and learning. God bless you all. Nice. That reminds me, I watched Muppets Christmas Carol in Orchestra the other day. What a classic. And little tiny Tim, when he was like, God bless everyone. Honestly, it melts my heart every time. But moving nicely on from my random escapades. Thanks, Lisa. Today, we are focusing on autism, but potentially this could be affected to any neurodevelopmental or neurological or mental health conditions. Yes, from Canada. Brilliant. So here's myself. And if you're wondering, my background is I'm studying neuroscience. So I have recently been learning this stuff. It should be noted, I'm not expert when it comes to genetics, merely someone who is very passionate about it. But I am potentially an expert in neurodivergence. Don't want to brag. But when I'm trying to mix those two together and think, how could it relate? So I am going to be simplifying a lot today. Not just for your benefit, but also for mine. If there are any kind of adjustments you want to say, please feel to let us know. So away we go. All right, as always, our team are majority neurodivergent, so experts via experience. We are talking from our own lived experience as well as professional experience. You got myself in the chat today. You've got April as always, and we got Izzy. You're welcome to ask as many questions as you like, and we'll do our best to reply to them as we go along. But the last one that is currently online now is teaching neurodivergent students. You also may have noticed if you went on our Eventbrite recently, we've uploaded a load of new events for next year. Really excited. I've been listening to what everyone has wanted and have been tailoring the content to suit what people have been asking for. So do sign up to those ahead of schedule if you're able to. But the future of neurodiversity. Now, by those little emojis, you may have an idea. Scissors and DNA. Obviously, this is super complicated, but we don't need to be experts in this in order to have an interest. And as people who are, a lot of us are ND, it's probably right that we know what research is happening in the background. That is the lens we're going at today. We are going to explore, if successful, what is CRISPR? It sounds some knockoff crisps from Aldi. How is CRISPR aiding neurodivergent research? Neurodiversity, of course, is the diversity of humankind, including the cognitive variations that we all possess. Can CRISPR, which we will learn is editing DNA, can it truly go side by side? We're going to be looking at treatments for neurodivergence. This is controversial because the neurodiversity movement in its kind of pure essence is we are fine just the way we are. We do not need treating. Treatment sometimes sounds like we're trying to get rid of something. But it could be used in another form, that making someone's life easier, more comfortable. Could we not, for instance, get rid of autism completely, but just get rid of the key limiting factors which can make some people's life very challenging? Could it be that is optional, that people could have, they could opt in or opt out? These are questions which are being raised right now. Ethical issues, if we start cutting away DNA, where does this end? Sounds a lot like eugenics. Hitler would have loved CRISPR. Controversial take, but It does sound scary, doesn't it? The fact that we could literally change someone's personality. We could change. Yeah, Lisa, that's your concern. And it is a concern. We have to know it to research it. And lastly, how could it benefit neurodivergence if we nurture it, if we have the right support in place? Could this be an amazing thing which cannot hinder genetic diversity, but allow it to really flourish? Let's find out. Some new terms, because I appreciate this is a subject which is not familiar to many people. CRISPR is, in its short, gene editing technology. It isn't future, isn't sci-fi, it's happening right today, where you can edit someone's DNA. You can take bits away, you can add bits, you can alter bits. There is so much you can do with it. A genome, this is all the genetic material in an organism. Think a nice bit packaged up. There you go, this is everything of who I am. Then pluripotent stem cells. These are stem cells that can become different cell types. When we are born, when we are just developing in the womb, we have unlimited possibilities. That little stem cell, could become an ear. That stem cell, maybe a lung. It could be anything. But if we were to use that same kind of cells, we can program it to be whatever we want it to be. Obviously, controversial. It used to be that people thought we had to harvest it from babies. But now we realize we can gain it from other areas. And then we have nucleolase. That's an enzyme used to cut DNA. Though I was showing my trusty scissors today, which I've been using for wrapping presents, it's not like we're actually cutting DNA. We're using an enzyme which kind of attaches on and cuts it that way. But it's not cutting in the traditional sense. Now, a quick question before, because I'm taking a lot for granted already. How familiar are you with terms like autism and ADHD? Okay, good, or bad. And this will just know so I can see where to pitch it. Okay, good, nice. This is good. I'm not going to spend my time overly indulging in that area. I'm going to predominantly focus on CRISPR and assume that most of us are aware. If you're not sure, just let me know. Now, what is CRISPR? I put some packet of crisp there because that is the first thing that always comes to my mind. Sounds tasty. But what is it exactly? Now, let me know your understanding. I've given a really basic understanding, but what is it really? It stands for, and this is where I'm going to butcher it, so sorry. Maybe I'm not. Hopefully, so actually, I would have struggled with pronouncing it anyway. But it is an acronym. I think it's an acronym, right? Yeah. We've got a form of gene editing, and yes, a form. There are multiple types. CRISPR does tend to be the easiest and the cheapest, and relatively quite new. It is an exciting place to begin. Brand new for me, don't worry. Okay, to cut out genes, a tool to edit genes. Think of it a bit like this. You know when you're writing a document on Microsoft Word and you spell a word wrong? Back in the day when you had a typewriter, if you made a mistake, you made a mistake. Rip the paper out, screw it up, try again, or live with your mistake. Now, with gene editing, it's like using a word processor. If you do the writing, you can actually say, you know what? I want to change that, copy it, cut it, delete it, paste it, change the font. You can do nearly limitless things. Editing has been a godsend for word processing. But what about when it comes to people? If you know your child is going to have a disability early on, would you want to edit the genes to remove that? It sounds good in principle, but then we bring up this question about what counts as normal and healthy. If this technology had existed less than 100 years ago, I guarantee you we would have edited the hell out of autism, get rid of it completely because we saw it as a negative. If you were gay, I guarantee you they would have edited the gay out of you. I don't know if it works like that. But the reason we don't do that today is because we've grown, we've developed, we understood this isn't a lot of these things which you would consider an illness is not an illness. It's just a different way of being. It's who you are. We do run the risk of pushing this technology faster than it is able to learn to walk by making these changes when we are still having a debate on what is healthy, what is normal, what is diversity, what is brilliant. Yeah, human rights. So you can see how we go. Here's exactly what it is. Is a tool, could it be a tool to understand neurodiversity? So here you've got your DNA. This is your genetic information. This is everything of who you are. Your DNA is all over you. And CRISPR is a molecule. A CRISPR molecule finds a precise location in the target DNA. So you've got your DNA here. And let's say this section here, that is the section we want to remove, get rid of, adjust. And now our DNA has like little markers all over it, like they're in like letters, and they're all different proteins. That's how they're spotted. Then we have a molecule that we kind of use in order, and what it will do, it will seek and find. Think of it when you do Control F on the computer. If it's there, it will find it. And what it does, it cuts through it, and then it will enzyme cut the target DNA at the point found by the guide. And then it will edit it by adding when the DNA is repaired. So we say, cut out the bit we don't want, put in the new bit, and then we're good to go. That's it in a really simple form. Oh, DNA is massive, by the way. We are talking really microscopic. And we know that, okay, when we did the human genome and we learned about exactly where we are, and we're still learning about autism and genomes, the idea, if you get the right bit, does exactly what you want. But we are operating on a tiny scale here. So that may be unforeseen circumstances. I think back to the example of Phineas Cage, when he was working on the railroad, there was an explosion, a big pike went through his eye, through his brain. He lost a bit of the brain, but he was alive. He seemed to be fine. And then we later find out he wasn't fine. Having that bit of the brain missing really affected his personality. But we didn't realize that at the beginning. Could this be a new example where we're editing DNA, seems fine to me, and then we realize there's problems later down the line? Then who becomes liable? You can see there are so many ethical decisions and dilemmas here. I'm not doing today in order to scare you. It's more about just thinking about the possibilities. Now for a CRISPR quiz. I'm sure everyone was excited to have a CRISPR quiz today. Super simple. All you need to do is let me know, what is CRISPR? Now, fingers crossed, you know this by now. Is it a cooking technique? Sounds like it could be. A tool for gene editing, a mathematical formula, a type of computer program. Yes, correct. All right, easy one to start us off with. CRISPR and neurodiversity. What is the connection? What joins these two seemingly unconnected things? One is a type of gene editing, another is the way we process the world. Okay, we've got by diagnosing them, yep. By computer programming, yeah. It's not related at all. Oh yes, developing treatments through understanding genetics. Absolutely. Yep. How it connects is the more we understand about genetics, the more we're able to support people. We aren't talking about a gigantic cleanse of all forms of diversity to create one super race. Easy, Hitler. What we are talking about, how can we make people's lives easier, more supportive? How can we allow them to be the best version that they can be, regardless of what society's expectations are? So you can see how, depending on your viewpoint, this can go extreme or really optimistic. Now, I think most of us can agree when it comes to mental health illnesses or diseases, this sounds like a no-brainer. But it's when we come to things like neurodiversity, where I'd say the ethical decisions really kick in. So CRISPR in ND modeling, this is it, super, super simple. First of all, we need to find the autism gene, for example. We should all know by now there isn't an autism gene. Autism, it comes from a number of genes and also environmental. We first thought maybe there is like this particular really rare gene, when actually what it looks to be is autism is many common genes. Autism genetics is really common, but it's like the sheer amount of them, and that's why it's different for everyone. Right off the bat, while we possibly could find a particular area for an individual, that doesn't mean it would be in the same place or same location for another person. That is our first problem. Create models. Edit genes using CRISPR. We can create models and we can see, okay, now we understand what the model looks like. We can choose what to keep, what to take, what to move. Naturally, you wouldn't want to be the guinea pig, would you, who has to do this first? But if it's a tried and tested method, which works the same in everyone, this could be great. I don't think we're dealing with that here when it comes to neurological differences, though. We want to study effects. We want to observe brain changes. Let's say we get around and we're like, how does that affect someone? And does it affect different people differently? And is it safe to do? Then we want to observe changes. So is the brain now operating differently? And explore treatment, develop treatments based on findings. To be honest, a lot of this we could probably do in mice and rats. And we wouldn't even dare do it on human trials for crazy amount of time until it is safe. But it's still something. And the next question is, do animal models really represent humans? Well, there are a lot of similarities, and it's not a bad starting point, but it doesn't tell us everything. But for instance, we roughly know how a rat thinks. We've observed them for ages. If we change its DNA and it starts thinking differently, then we know it had worked, whether correctly or incorrectly. Here is an example of CRISPR in Alzheimer's. Obviously, Alzheimer's and autism is very different, but there is some overlap. This is a very complicated image, so I don't expect any of you to take it in. But essentially, there's lots of different kind of genes, and it will be able to pinpoint the exact one, cut it, rewire it. And essentially, in the brain, when you've got Alzheimer's, it's part of the brain kind of deteriorating, and then it spreads almost. If you can cut that bit out, then Alzheimer's begun, potentially. And you think, if we're already doing this in Alzheimer's, which, let's face it, is unanimously terrible, could a similar thing be used in autism? Now, this brings us on to challenges and ethics. I just want to get a feel of the room. How are you currently feeling about what we're talking about? Are you very uncomfortable with the subject? Are you neutral, is what it is? Are you very comfortable? Or are you like, nope? All opinions on this are completely valid, by the way. Okay, we've got some neutral, we've got some comfortable. Honestly, I'm a little bit shocked. I thought there would be some people who were like, hell no. Can you be both? I think for me, I'm uncomfortable, but also excited. I've got that kind of like curious cat personality where I know it's probably bad for me, but I also want to see where this possibly could go. Now, what are the main complexities? Regardless of whether or not we like this, is this even possible? So is it the genetic complexity? The fact that all our genes are so different. When we are talking about neurodiversity, while we talk about individual conditions, there's no such thing as far as we're aware of a dyslexia gene, an ADHD gene. We are just loads of different characteristics. And diagnostic labels are a precursor or a remnant of psychology and don't really represent neuroscience. Then we've got ethical concerns. This is morally, is it correct? And thinking about it on a societal level. We've got unintended consequences. Are we creating the next Frankenstein? Hopefully not. But you could see, are we interfering with are we playing God, essentially? Individual variations, knowing that, again, everyone is so different, and society and regulatory challenges. Even if this was possible, you can know for sure that, say, Texas is going to be like, nope, not doing that. Sweden might be all for it. Depending on where you are in the world, I think there'll be very different stances, and it could create an underground market. Genetic complexity wins. Yeah, it is not a simple task, but another little kind of ray of hope, if this is something you are interested in, AI is ridiculously good at solving complex problems. So as long as it isn't impossible, AI stands a very good chance of being able to work it. We already have the genome, like kind of the map of humans. Now it's just about finding it out. Naturally, you've always got to look at the human studies. Are we taking people from all different backgrounds? But we do have the building blocks already. All right, for next one, we've got CRISPR and neurodiversity, ethical challenges. Lisa says, I think there are about 35 genes that code for autism. I have definitely seen that, Lisa. Truthfully, I don't think we know yet. If we knew all the genes for autism, I think we'd be much further along in our development. I think we have some areas which are real hotspots, which have definitely shown for some individuals. But truthfully, I think our definition of autism is too broad to really nail it down to a particular set of genes. Oh, no, there are way more genes than 35. I think 35 are just the ones we've got our eye on. Okay, let's have a look. We've got genetics improve neurodivergent knowledge. So the more we know about genetics, the more we can support people. Sounds good to me. Ethics are key in neurodiversity research. Some of you didn't agree with that. I do think we always have to question, is this like I want to say Jurassic Park quote? I don't know. We wondered if we could not if we should. And it is that question, just because we can do something doesn't mean we should. Genetic treatments for neurodiverse is debated. Some of us see it as a disability. We want to get rid of disabilities, ideally, so no one wants to be disabled. But do we want to get rid of cognitive difference and variations and people? No. So you can see, depending on your personal viewpoint, this might be very controversial to not at all. Research influences ND cognitive diversity. Actually, I just want to make one little clarification. When I said we don't want disability, when people have disability, of course we should be supportive and encourage them, and everyone has a place. But obviously, if we can avoid suffering from an individual or take it away, of course, I think most of us would want to. That's just want to clarify, because you can already see how this is a very grey area. Fair access to ND treatments needed. So we've got a few little things to think about here. Let's move back to Alzheimer's now. And what do Alzheimer's and autism have in common? Now, I have autism, and do I have Alzheimer's? I hope not. To be honest, it runs in the family. But are they necessarily connected in that sense? Probably, maybe not. But are there connections on a genetic front? After all, many geniuses are autistic. I try. No, but then there's also many not, like autistic people are so wide varying. So we've got both are genetic. Yes, Alzheimer's and autism are both genetic. Diagnosed at similar life stages, not at all. Autism is diagnosed very early on, where Alzheimer's is typically later on in life. Both affect brain structure. Yep, they absolutely do. They both alter how the brain processes, and not caused by environment. No, they can be. And that's the thing, autism does have a genetic component to an environmental component to it. So as you can see, if we were to do it against studies we've already done, there are overlaps, but there are also areas that just do not line up. What are your biggest hopes for CRISPR gene editing in neurodiversity therapy? And I think therapy is a nicer word, right? Because we're not talking about removing, we're just saying supporting people to live their best lives. On here, just move up and down to which one you think is the biggest hope to the least hope. Okay, number one, a deeper understanding of neurodiversity. Yes, the more we know, the more choices we can make. I always think that it should be nothing about us without us. These really big parts of research, we should be in the driving seat. We shouldn't just be allowing it to go off in the background. Potential treatments, yep, I like that. Remember, treatments do not have to mean ridding. It can just be supporting or helping people live independently. And then we got personalized medicine. What do we mean by this? Let's say you've got ADHD at the moment. You'll essentially get some generic medication, probably a stimulant, and see if it works. Oh, did it work? I'm not sure. And if it doesn't, try something new. With personalized medicine, what you're taking is the exact right thing for you, and it's going to have the exact right implications and impact that you expect. With autism, while we do not have autism for medication, we do use medication in order to support some of the co-occurring conditions that go along with autism, such as anxiety or depression, conditions like those. So you could argue that, yes, that would also benefit as well. Now, here is a nice blurry picture of Steve Silberman and a quote from his phenomenal book, NeuroTribes. Neurodiversity may be every bit as crucial for the human race as biodiversity is for life in general. What a quote. What this is saying is thinking differently is as crucial to the human race as biodiversity is. So biodiversity is all our nature, our animals, our everything around us. And we need all of this to live unanimously. We are destroying our biodiversity, and as a species, we are suffering as a result. If we destroy human diversity, we will also suffer just like we are with the destruction of our biodiversity. Yeah, I personally love that. Who can say what form of wiring will prove best at any given moment? And this is really true. The way that we have created our world, it's that a certain type of wiring is the preferred way, and hence that creates disability for those who do not have that wiring. But if we allowed people a bit more flexibility, you'll notice that different types of neurology have different benefits and perks and strengths depending on different scenarios. So with that in mind, if we were to edit people's DNA, we potentially could lose this diversity of what makes us special. Are we going to be repeating the same mistake that we're doing with our biodiversity? Definitely food for thought. Moving a little bit along, have you heard of
amyotrophic lateral sclerosis?
Let's just say LLS. And so, no, LLS is a fatal disease that causes motor function impairment. In contrast, conditions like autism and ADHD are related to lifelong differences in the brain. These are different conditions, by the way, very different. One is how your DNA is wired naturally, and another one is when your connections in your brain are unhealthy. So say you've got your muscle, here you've got your neuron, you've got your synapses, you've got your dendrites, you've got all your good stuff here. Now in ASL, this harms the motor neurons, making the muscles weaken and can become paralyzed. So essentially, your cables in your brain, which kind of get information through A to B, are now dead. So this is something which potentially CRISPR could be really useful on. We could edit out the dead neurons or the dead genes and make sure that you are healthy. You can see there is maybe some overlap, but there's a lot of things that are quite different as well. In the UK, we tend to call ALS motor neuron disease. Oh, thanks, April. Ugh, I feel silly. Yeah, you are totally right. I should have used that. Appreciate that. While we know there is really interesting research happening, it doesn't necessarily apply to neurodivergence. On this screen, you've been given a load of money, and I want you to tell me, where would you invest the money? So we've gotten for autism, ADHD, dyslexia, and dyspraxia. These are things which are theoretically possible, but still have a lot of develop to go. We have autism, CRISPR on social genes for therapies. The fact that we find things socially awkward and can be cripplingly destroyed with anxiety. Should we look at ways to support and to minimize that? ADHD, what about the attention gene? If we could get rid of the inattentiveness and allow people to concentrate more, would that be a good use of time, research, and funding? Dyslexia, CRISPR on reading genes for learning. So when people struggle with learning and the way they interpret written information, we could completely remove that and make sure that people can read nice and simple. Dyspraxic coordination gene, so allow you to be more coordinated. Do you know what? I'm struggling with this myself because, let's face it, a lot of the negative characteristics of conditions, they can suck. They can be really challenging and they can make life really difficult. But if we get rid of them, what's left? And I think neurodiversity is about the fluctuation of abilities. It's about the skills that are really high and the skills that are not so high. And I wonder that if we crop out the low skills, are we creating superhuman soup, or are we ridding diversity? Maybe highs and lows are both necessary. You can't have your highs without the lows. Again, more food before, no particular answers as of yet. Now, CRISPR requires specific ways for tricky conditions. The ones we're talking about, ADHD and autism, are tricky. And there's a few reasons they're tricky. Let's look at the spinny wheel of trickiness. Delivery, how it is delivered is going to be different for everyone. And let's face it, it is invasive in some forms. And then we've also got precision. How accurate is it? Let's say you want to cut off this bit of DNA, but you accidentally cut off this bit of DNA. It may not look like a big difference, but it could have unforeseen circumstances. We've also got safety. Is it safe? Is it reversible? If we don't like it, can we turn back the clock? There are a lot of other things to consider. Let's summarize now. Ethics in neurodiversity gene editing, consent, informed for vulnerable individuals. Those who would probably most benefit from this type of technology are probably those who are the most vulnerable in society. Who is making this choice for them? Do you have to be over 18? Do you have a certain level of IQ? Are your parents able to make that choice for you? Stigma. If we continue down this route, are we going to be undoing all the advocacy we've been doing for years and years? We have finally made people realize that being different is an advantage in a lot of places in society. There's still work to be done, but we have come a tremendous way. If now we are saying, actually, if you choose not to be fixed, you're the problem, you could see how this could send us back like 50 or 60 odd years. Then we've got equity, ensure fair access and benefits. Let's face it, this is going to be expensive, and it means that people from higher income backgrounds are going to have higher access to genetic editing, and may have an even greater unfair boost compared to the rest of us. Complexity, we've got to appreciate this is not an easy thing to do. This is real high-end stuff. And long term, we do not have the studies yet because this is far too new to see how this will affect us later on. I'm going to talk about bananas now because bananas are lovely. Bananas are genetically very vulnerable because we used to have lots of types of bananas, and then we found this really delicious banana. Oh my God, what a great banana it was. So we started to clone the banana, and we use its genes and would make copies of copies. Pro, no seeds, really easy to eat, very easy to create the optimal conditions for growing. But now, basically, every single banana is vulnerable. All it takes is for one really bad disease, and if it catches on one, it's going to wipe them completely out, and then we've lost that banana forever. You could see how that could apply to other areas. Same with like grapes that have no seeds in. They're great for us. I flipping hate grapes with seeds in, but it's terrible for diversity because we're just cloning the same mistakes, and it makes them really vulnerable. Oh, Peg says, what are the medical concerns of newborns from mothers that were gene edited before the child's birth? Now, that's a good question. I don't know the answer to that, and I don't know how possible it is to edit genes within the womb. I would have to do more research on that to see if it's even been done before. But it's definitely, there's possibility that it's something that could happen. All I was saying is I'm not ruling it off the table. Our key takeaways today is CRISPR edits genes effectively. It does actually work, okay? There's been no negative repercussions that I'm aware of or that I could find. It does actually work. Studies, and it does treat brain conditions. While it hasn't been done on neurodivergence yet, it has been done on conditions which are unanimously bad, and getting rid of a particular gene could benefit someone's life. It has also been studied substantially on animals. Overcome stem cell limits. We know that stem cells are used in order to grow new organs and things, but we are limited at being able to get these stem cells. This might be another route around it. Precision precisely changes genes. If you want to change your genes at the moment, this is really precise, but does it need to be even more precise? And edits brain condition genes like in autism and schizophrenia. Possible, but debatable whether or not this is something we should even entertain. Regardless of whether or not we're going to entertain it, if it's possible, people always do it. Can it work on schizophrenia? I would say yes, Louise. It would, in theory, work on anything where we're able to pinpoint it down to a particular set of genes or genome or genes. In theory, yes. I appreciate today has been a real kind of like introduction because this is a complex area and it would need someone far more experienced in this to really give it justice. But I hope this has maybe introduced you to the subject and tickled your interest. There's a really good book that I started reading called The CRISPR Generation, the story of the world's first gene-edited baby. You might have heard of the term designer baby. It could literally be a thing. I want to have a baby with brown eyes. I want to have one that's not too fat. I want one that grows up to be really handsome. I want one who's really smart. If we truly can design our own human, like you can do on The Sims, we may end up with the same person repeated. Or let's say you're a huge Taylor Swift fan, you might be able to order your very own Taylor Swift baby. Scary, but it's good to think about these while alongside developing the research. So yes, the CRISPR Generation, the story of the first gene-edited baby, is a really interesting book. Not really related to autism, though it does mention it, but I still think it's a useful book. Where to access this medical procedure? It's not open to people, not for a long time yet. So yeah, do not be looking at the yellow pages and thinking, I'm going to find myself a CRISPR editor. They don't exist yet. A Crack in Creation is also great, written by Jennifer Duggar. Well, thanks, Lisa. I'll check that one out. This is not related to gene editing at all. But if any of you are neurodiverse and quite happy with your genetics and do not want to be cutting them up left, right, and center and just want support in the work environment, we do offer workplace needs assessments. Now, our next webinar, and I believe we only have 2 webinars left of the year, is Neurodiversity and VR. This is really interesting. This is also about the future, but a completely different future. Not a future where we change our genetics, but a future where we find alternative ways to support people. We know that going into a new environment, a shopping center, going for a new job can be absolutely terrifying. But what if you could do virtual simulations in advance, which can help you get more comfortable with those environments? We already do it for phobias, like if you're scared of spiders, you could have a spider in your head. Don't worry, they do it slowly first. But this is an alternative future that we may also look at. And I just want to show you all real quickly while we've got a few minutes about all the amazing other workshops that we've got coming up, because I've been adding them all last night and I'll finish adding a few more today. Here we are. Look at all of these. Do I have autism, ADHD in 40s, predominantly inattentive ADHD, Savant syndrome, looking at reading disorders? We have got so many great ones for next year. They are well in advance, but if you sign up before, it will show that there's an interest and it will mean that I can keep creating these and keep putting them out to other individuals. So some really interesting ones. Do sign up to these. And this is just on our Eventbrite. And here is our YouTube channel. If you want to click and subscribe, and you can see all our previous ones. If you found today useful, check out our autism neuroimaging, autism neurobiomarkers, and autism origin webinars. They also go into how genetics and autism are closely aligned. And here are our contact details. Whew, that was a lot of information today, and I appreciate everyone being so mature about it because this can be a bit of a hot topic. So thank you, everyone. And I hopefully will see you next week for our penultimate 2023 webinar. Cool. Oh, thank you, everyone. Really appreciate it. Nice. Bye, everyone. Have a good rest of your day.